Good afternoon, ladies and gentlemen. It's lovely having you in this room. And this is going to be a very nice session, applied real-life plant pathology for food security solutions. So we're looking forward to this. Welcome, come inside, and we have 5 exciting young dynamic speakers who's going to share with you their research. And at the end, we have Serge that's going to give you an overview of our journal and food security in the bigger context. So Serge and I will be chairs, and as you know, Serge is the chief editor of Food Security, the Springer Journal, and I'm the chair of the Commission of Food Security. And I hope you're all going to come and join us and participate in our society's Food Security Commission. And with that, this is the program for today, and we're going to start straight away with our very exciting speakers. We have 5 of them, and I'm going to ask Nina to come in and start with her presentation. And Serge is just saying a word while we're uploading her talk. Okay.
Hi, good morning everyone. So yes, you are at the right place. That's a food security session for sustainable food systems. Welcome. We shall have 2 parts in this session. First will be presentations, 20 minutes each. Sorry for the speakers that you have only that time. After 15 minutes, I shall begin to become sort of agitated, meaning to say, I'll try to not be agitated before. and so, but don't stress. Lise and me, we are your, we're not supposed to be, but we are your friends. We are trying to make it so that your presentation goes okay. And then, so please don't stress, please, speakers. And then unlike me. And then we have a sort of forum. I will be presenting to you our ICPP journal for security. And then I will be looking for feedback from you on this, because it belongs to you, all members of ICPP. So with this, thank you very much.
Thank you. Thank you to the panel. Good morning, everyone. I'm Nina Ockenden-Powell. Very excited to be here, so thank you for having me to speak. I'm a senior research associate in the School of Biological Sciences at the University of Bristol, where I am one of the Connected network managers. And Connected is the community network for African vector-borne plant viruses. Today, I'm going to tell you a bit about what Connected is and why we need it, and how Connected has been supporting vector-borne plant virus research capacity through research, training, and engagement, and how Connected may be now presenting a new model for developing research capacity. Oh, I seem to have missed a slide here. So originally, Connected was one of 4 vector-borne disease networks funded by the Global Challenges Research Fund via the BBSRC, that's the Biotechnology and Biological Sciences Research Council, part of UK Research and Innovation. The call that funded these networks had the aim of supporting a complementary community of vector-borne disease networks to deal with the challenges of vector-borne diseases in low and middle-income countries. Connected was the only plant-focused network of those. So the objectives of Connected, which echo the key objectives of the call, were to build a sustainable and long-lasting network of multidisciplinary international scientists to address the problems of vector-borne plant viruses, to run a series of activities to promote and embed interdisciplinary working, to strengthen research capacity, capability, and methodologies, particularly focusing on the vectors of disease, to prime fund a range of new projects to lead to more competitive, collaborative, cross-disciplinary research proposals that link the UK with sub-Saharan Africa and deliver impact, and to capacity build via improved communication and collaboration and through events in the UK and in Africa, and specifically to develop early career researchers because they are the future. Also to support collaboration between researchers in the UK and sub-Saharan Africa, and to provide legacy benefits by facilitating the opportunity for further funding for projects that bring the greatest impact to the region. So we distilled these objectives down into our core mission, which is to build multidisciplinary research capacity to tackle crop viruses spread by insects, to ensure fair, sustainable, nutritious food production for all. And we do this through research, training, and global engagement. Connected began in 2017, led by the University of Bristol in collaboration with Newcastle University. Connected is currently funded by the Bristol Centre for Agricultural Innovation within the School of Biological Sciences at the University of Bristol, which will end at the end of September this year, so next month. So the photos here show our leadership team. We have Professors Gary Foster and Neil Boonham. Gary's from Bristol, Neil is from Newcastle. Neil is just sitting over here this week. So they set up the network. Gary has recently retired, although he still remains involved. So Dr. Andy Bailey, you can see on the far right there, he's stepped up into the role of director. He's also at Bristol. And then we have the network team. So from left to right, we have Dr. Deanne Hurd and myself. We job share the role of network manager. So we plan, implement, and oversee the phased program of activities. We build collaborations and partnerships in the UK and overseas. And we monitor and evaluate our impact. And then we have Heather Child, our Senior Communications Officer. Communications has been a really, really important part of this project. And we have Krista Muller, our Executive Assistant. Connected is governed by an expert international management board of around 20 senior researchers and stakeholders in plant health, molecular diagnostics, and impact in agricultural systems from across Africa and the UK, most of whom have been with us since the very beginning of the project and have given us their time and expertise for free. The management board is chaired by Dr. Lava Kumar, who's head of the Germplasm Health Virology and Molecular Diagnostics units at the International Institute of Tropical Agriculture in Nigeria. He's also here this week, so do seek him out. For the first 5 years or so, the management board was chaired by Professor Nicola Spence, the UK Chief Plant Health Officer. So why do we need a network like Connected anyway? Very simply, vector-borne plant viruses decimate a range of staple and nutritious crops around the world, compromising food and nutrition security, particularly in vulnerable regions. In 2016, our funders conducted a survey which highlighted the main gaps in the research and training landscape. And it identified strategically important priority areas for building research capacity, shown here in a figure taken from that report. And the top 6 of these became our top priorities for our activities to focus upon. And what was clear from these activities, and what became clear to us very early in the project, was that the network had to actively bring together expertise in plant pathology and entomology to effectively build capacity in vector-borne plant virus research. So that became our interdisciplinary niche. And with this clear interdisciplinary focus, we set about designing a phased program of activities guided by a theory of change to ensure that all that we do focuses on the impact that we want to achieve, so increased productivity, food security, and income for smallholder farmers. And that this is all underpinned by a robust monitoring, learning, and evaluation framework that tracks and reviews our progress. One of our key objectives is to enable our members to successfully apply for research funding. So to do this, we have Pump Prime funded a range of short-term research projects to generate proof of concept results or pilot data for use in larger funding applications. We co-develop and run interdisciplinary training courses and provide training grants for our members so that they can develop their knowledge and skills in vector-borne plant virus research techniques and methods, and so that they can develop their professional networks and ultimately their careers. We primarily focus on early career researchers, as I mentioned, they are the future, and we offer our opportunities in a gender-balanced way wherever possible. And then we run engagement events, including conferences, seminars, and workshops, online and in person, bringing people together to share knowledge and to develop collaborations. And we also provide a wide range of resources via our website, including laboratory protocols, training videos, and awareness-raising infographics and material. And we keep our network engaged and interacting via a range of communications activities, including regular monthly e-newsletters and social media. And this is where we are right now. So we have nearly 1,700 members across 96 countries. I'm sure you'll agree that with us that we consider Connected to be a truly global network. So in terms of our research pump priming, this infographic summarizes the whole portfolio that we've funded. 20 projects involving 55 researchers from 33 institutions across 14 different countries and involving 11 different crops. And these have gone on to secure over £300,000 in additional onward funding. Over the last year, we've been able to take forward translational activities from a handful of these projects, working on things like new biopesticide development, new surveillance and molecular diagnostic techniques, and the provision of practical advice for farmers for dealing with vector-borne plant viral crop diseases. In terms of training opportunities, we have trained around 140 early career researchers from 23 countries. And through our monitoring, so involving online surveys before, after, and at 6 months follow-up after our events, this has showed that our trainees have shared their knowledge and skills with around 1,000 of their colleagues in their home institutions. So this represents a really wonderful organic cascade of the new interdisciplinary skills that they've gained via Connected. And if you visit our website, you will see a range of publicly available materials, and if you join, you'll find a whole lot more, such as our new online training modules in virus and vector diagnostic techniques. These are all freely accessible once you become a member, and membership is free as well. So, yes, the Connected Network has been successful in building a very large international membership, particularly amongst early career researchers in sub-Saharan Africa. But has it achieved its goal of increasing vector-borne plant virus research? Has it integrated expertise in plant pathology and entomology? To answer this question, we've been working with Professor Bruce Cronin of the Networks and Urban Systems Centre in the University of Greenwich in the UK. He and his team have been conducting a social network analysis of Connected, looking at how it's evolved over time. And I can share a few preliminary figures from this. So they've been looking at patterns of recruitment. So you can see the red line is the cumulative membership over time, and the blue highlights the spikes in members joining, which can be mapped onto our different activities. They've been looking at expertise recruitment. So when members join, they tell us about their different expertise and knowledge areas, and so we can see how those different areas contribute and fluctuate over the life of the network. And they've been also looking at determinants of engagement, and I apologise, this is not a particularly clear figure, but it's to illustrate how we've been looking, how they've been looking at how our members interact via attendance at our events and participation in our funding calls over the years. They've also been looking at how social media interactions are linked to connections between members via our events, and whether these indicate a higher whether these relate to higher order indicators of collaboration such as publication. So yes, the study is showing that we have been indeed successful at integrating plant pathology expertise with entomology. The network has developed a core of related technical expertise and knowledge capacity alongside distinct communities of expertise that are fluctuating over the life of the network. And we're now using this to develop a new model for successfully building research capacity, which incorporates the appropriate conditions with which you set up a network, including funding, the governance structures, our motivations, communication styles, and the sequencing of events that enable people to come together and connect and then disperse and share information in a kind of hourglass model. And all this being facilitated by a phased combination of in-person and online interactions. And the way that this seems to be able to build research capacity is by enhancing the ability of the community to produce outputs, such as the ability to publish, to generate new innovations, and to collaborate or work successfully across interdisciplinary boundaries. And we hope to publish this study later this year, and we really hope that this could be adopted by funders and other organisations wanting to promote research capacity as an effective approach. Now, I'd like to just share a video, one of our awareness-raising videos that we've produced. One of the most wonderful things about working with Connected is seeing how our members have taken the new knowledge and skills and are using it straight away in their work and sharing it with their colleagues. So this is about 1.5 minutes long.
Connected is a global, multidisciplinary network. We connect around 1,500 members in 82 countries, researchers whose work is crucial to global food security. We link plant pathology and entomology to tackle plant diseases that are spread by insects. We grow technical expertise and knowledge and increase international collaborations. We've trained more than 100 early career researchers from 18 countries who have shared their skills with hundreds more. We funded 20 new innovative research projects on 11 different crops, making brand new connections between institutions in the global north and south.
Basically, with all the new techniques I've been able to gather, I think it will really help me to take my career, my research, my profile as a scientist to a higher level.
When I'll be back, I'll be able to share this information with my team that I manage. This will help us to collect and improve our work as we are working on the viruses and other challenges in Tanzania.
So this video really highlights to me the need for onward support for networks and approaches like Connected. I mentioned that our current funding is ending at the end of September, so our next steps, our current activities are to ensure the sustainability of the network. And to do that, we are working towards transferring the network to a new host organisation who will continue the work that we've started. I can't identify the new hosts as yet, but we have an excellent first choice and an excellent reserve choice. So as such, the network will continue with the ethos with which it has been set up, and we see this as a huge legacy achievement. Thank you.
Thank you so much for this wonderful, outstanding, superb talk. Are there any questions? Yes, please.
Okay, thank you for your question. So the first, the spikes in memberships joining, those huge spikes to start with were our launch conferences. So it was very important for us to initially bring together in person our developing members. So we ran launch conferences in the UK and in Uganda. So when we were targeting different organisations for new membership, we had these spikes in members joining, and related to those conferences, we timed it with the funding calls that we were about to issue. So that drove a lot of interest. We wanted to get people excited about what we were doing. We did a lot of communications activities, sharing information, so we got these huge spikes there. The others, going further along, were our in-person training courses that we ran. So we ran these open competitions. Again, the communications, the promotion of these was really important to drive interest and excitement, and we had a huge number of applications. Even with providing awarded places on other people's courses, we had a huge number of applications for a residential bioinformatics course that was run at SLU in Sweden. So all these activities, you excite people about what Connected is doing and provide them with the opportunities to develop their career in the areas that they need to. And we've responded very much to what our members have told us they've needed. So I hope that explains that. I can show you more detail afterwards if you'd like. In terms of geographical spread, yes, initially Connected had the sole purpose of focusing on sub-Saharan Africa. So we focused on partnership organisations there that some of our management board were already connected with, but then organically over time and through our various activities and promotions, we've had an organic growth of the network across the globe. I think it was a year or so ago we did a bit of a review of where our membership was, and we were absolutely, we were so surprised to find members all over the place. So it just shows, I think, the value and the need for this sort of approach.
Any other question?
Yeah. Okay.
I have one, but any other question? We have still time. This is a unique talk that I heard.
Thank you.
Well, I have one which is not very scientific. You mentioned one CG center as playing a key role in but what was your audience? Who are these people? Are they coming from CG centers? Are they coming from national institutions? What kind of ratio is that?
We have members coming from many different types of organisations, CG centres, National Agricultural Research System organisations, universities, a whole range.
Okay.
Yeah.
To my shame, I must confess that it's my first time to hear about this fantastic project, and it suggests that I'm trying to keep in touch with things, but I would advise maybe you may want to forward some notes to ISPP News, to the ISPP newsletter. It would be most welcome. And yeah, I am editor-in-chief of Food Security, so voila.
I'll come and talk to you afterwards.
I won't be too heavy on this, but this is a talk which speaks of the future of the next generation of scientists. It speaks of food security when we shall not be around anymore, and this is important. So I would like to thank you very much.
Thank you very much for your comment.
I think it's time for the next speaker. That's Dr. Lise ROTHMANN. Hello, please come. Don't be afraid.
I luckily did this yesterday, so
Okay.
I feel I'm already warmed up. Thank you. Okay, good morning, everyone. So I'm actually presenting on the behalf, I'm a whole person, of a colleague. So this is really outside of my wheelhouse, besides that I work on soybean. So I've got my crib notes here, and maybe if I didn't say anything, no one would have known. But I need to be fully honest if I'm going to have integrity. So, and the reason why my colleague is not here today is because she's expecting twins. Yeah, I don't think in the heat wave it would have been advisable either to travel. So today I present to you a quantitative and qualitative analysis of the rhizosphere microbial populations of maize and soybean as influenced by soil and plant genotypes. So this work was done by Karen Fenter, and it was done quite a bit ago, but it's still, I think, at the time it was really relevant because they were looking at the differences in the glyphosate tolerance and the influence of the soil types. Her group is the soil microbial ecology group. That is, the research lead is Professor Wynand Swart. And so this is about the importance of soil health. And yesterday we also heard a lot about, soil health means ultimately drives plant health. And if I listen to the keynote, I should say, not necessarily the legislation, but the outcome of how we have healthy plants. And yeah, ultimately then influences agriculture and our human health and livestock health. So we all know that the soil is a very dynamic system, and the microbial or the rhizosphere microorganisms can allow for a lot of activity for the nutrient transformation and element cycling. And they wanted to look if there's any impact of the GM crops on the rhizosphere microbial populations, and to see if there's any alterations in these environments that will then affect soil health. So their hypothesis was to look at root exudates of GT and BT maize cultivars, as well as the GT soybean cultivars. And this will then lead looking at to the qualitative or quantitative changes in the activity of the rhizosphere microbes. So the trial had two different soil types, a low clay and a high clay soil. And I'm not going to spend too much time on the details, but there were clear differences in the soil characteristics, as well as the organic matter and the silt percentage and sand percentages. And they had 8 different cultivars that were used, 2 of which were near isogenic families. And then for the soybean, they had 4 non-isogenic soybean cultivars, and these are all cultivars that are available to producers, or were available. Some of them may no longer be in use, depends on their popularity in the production regions. So they use different biochemical techniques to look at the microbial activity, the total fungal biomass, and they use some molecular techniques, DGGE and TRFLPs, to go and see how does this all cluster with cultivars or the soils. So let's have a look at what they found. With the maize, with the microbial activities, they had a greater activity in the low clay soils, and we'll see that this becomes really interesting because it contrasts with the results of the FDA results. So here there was differences between the GM maize and the conventional maize groups, but it was not a significant difference. There were just some tendencies that they observed. But with the ergosterol, there is much greater variation that they observed with the low clay amounts, and there is no pointer, but it's this side. And then the higher clay content, there was a tendency to have a reduction in the ergosterol. But then if you contrast with the FDA, there was a much more there was a significant difference in the activity, microbial activity, in the high clay content than in the lower clay content. So this then contrasts to the CLPP, where they had greater bacterial communities in the low clay content soils. So with the clustering with the DGGE, they saw that there was only clustering in the soil type and not the cultivars, which is interesting considering the fact that with the other technique, they found that the cultivars actually had differences. So they were contrasting results using the different techniques. Here again, just to show that the high clay content and the low clay content were those that clustered together. And then there's a group that's kind of on its own, but also all high clay content soils. Similar pattern that we can observe in the RFLPs with the high clay content clustering and then the low clay content. So I won't spend too much time on this, but you can see there was a lot of effort put into this research. So with the soybeans, for the bacterial microbial communities, there was greater soil bacterial communities that influenced then rather the cultivars, similar to what we saw in the maize. But there were no significant differences in the maize GM compared to the conventional soybean, unlike with the maize. Where maize, there were significant differences between the type of cultivar. There were no significant differences here between the cultivars. With the total fungal biomass that was detected with ergosterol, there was no significant differences detected, but ultimately the lower clay content had lower concentrations, and the high clay content had greater total fungal biomass. I'm also aware that with ergosterol, you're not only measuring your active fungi, you can measure those that are no longer living. So that is something to consider. So with the FDA, here again, there was a greater, there were more activity, microbial activity happening in the high clay content, so we don't have contrasting results here. We are seeing similar tendencies with the soybeans. Here again, we also have the clustering of the low and the high clay contents. So there were no significant clusterings or groupings with these. And again, with the clustering, the TRFLPs, we see something similar again. It's not, the clustering isn't based on the cultivar type, but rather on the soil type. So in conclusion, or in a discussion for this, it seems that the microbial populations of the maize hybrids do differ from the conventional hybrids, and this may have to do with the interaction that is occurring and the rhizosphere layer and these different cultivars, and we know that this is most likely to occur. Then the alteration in soil microbial activity caused by these cultivars compared to their conventional partners can also then change how could potentially change how the ecosystem's functioning within the farming system. And they also saw that with the soybean, though, there were no differences. And ultimately, they are looking then where future research could then determine or include yield parameters to see if they're actually making any economic impact for the producer. I mean, it's great to do the research and find the results, but ultimately we need to know what influence this will have on the producer's practices and selection of the cultivars to preserve the soil that we have. So the results obtained from the soybeans were inconsistent with the findings of the maize genotypes, and this did affect on the soil microorganisms. We also need to potentially look at the insert gene of those specific crops and look at the soil microbial populations. And the clay and organic matter of the soils were important considerations when you look at the characteristics of the soil specifically. So in conclusion, there was no significant effect of the CP4EP-SPS gene on the rhizosphere microbes, with higher microbial activity evident in the high clay content than in the low clay content soils. The results could not confirm that GT maize and soybean has a potential biosafety hazard to the rhizosphere microbial activity, and further investigations are required. And if you have any questions, please direct them to jeffreycarden@outlook.com.
Are there any questions?
You can try. I don't know how far I'll get.
There is one here. There is another one here. Just next to you.
Hi.
Hi.
It was very interesting. I don't know if this works. I was just wondering if maybe because the clay accumulates more water, that's why the microbial activity is higher. Do you know, just check that or so?
I will make a note. I feel like I'm back in like honors. In South Africa, we have honors. In Australia, you also have honors. I feel like I'm back in honors. I need to like make a note and go and ask my professor, who's now the presenter. But I will tell her to check about the soil and the water content.
Thank you.
I'm aware of it personally. I'm not sure if she's considered it. Yeah. I think also what would be interesting to see if these were produced under dry land or under irrigation, because that will make a very big difference as well. Yeah.
Other questions for Lisa? Yes, please.
Just a quick question whether the glyphosate was actually applied to control weeds.
Yes. Yes, they were. It was.
And the same dose between GM and non-GM.
Yes. Yeah.
Okay, thank you.
There was a hand raised there. We have time.
We can even make this a discussion. You know, we can get everyone involved. That's why we're here.
Not yet, not yet. Another question?
Maybe about the discussion. I think it's okay. So this was about GMO for BT and also glyphosate tolerance. So do you think that the results which were obtained for the GT tolerant germplasm can be also valuable for other future herbicides, because it's in some parts of the world glyphosate is already banned, and it may be so in some other parts. So I'd like to know if you think that there would be relevance for future and other molecules.
Yeah, I think the likelihood is there. Besides this research, our research group is going to start looking at the use of herbicides in the desiccation of sunflowers for Sclerotinia head rot protection. So it will be interesting to consider that, because it influences the cropping system. So in South Africa, they are rotating maize, soybean, and sunflower. So if we have GT crops with the glyphosate tolerance, and then we also would use other herbicides, it would be important to understand if that is going to occur. Yeah.
I have a quick comment to make, which is this. I know we are in France, and so it's full of French, and the French, they drink cognac. And they drink cognac, and therefore you have two types of cognac. You have the cognac 2 stars and the cognac 3 stars, meaning something. In statistics, it's not the same. It's not because, like I saw, a probability of 0.07. Now it's up to the experimenter, it's up to the scientist to decide whether that is significant or not. It could even be 0.01. It is all dependent, the probability levels are all in the hands of the experimenter. The only thing is that that probability level, if considered significant, has to be justified by the author. And so this is not, so these values, P00001 and 01 and 005, these are standards. It's outdated. That's the past. That was before. Now, it's in your hands. It's in our hands. We decide what is significant and what is not, but we have to justify this. It's important. Yes, ma'am, you can go. You may go.
Thank you very much. And let's go over to our second last speaker. And we have a replacement speaker as well. This is a replacement session. And I think it's Dr. Misiani, is that right? Okay, almost, one day, soon. Okay, and we are looking forward to your presentation on assessment of cultivar potato lines. And let me hand over you too. So nematodes, here we come. Let me just do your,
there we are. Thank you. Hi, good morning, everybody. I think the bar's already been set so high. Everyone who's been ahead of me has been a doctor, but anyway. I'm an MSc student pursuing an MSc in Global One Health, and my background is in zoology and biochemistry. However, I'm a trained nematologist. So you're asking me where I come from. So yeah, basically I come from Africa, in Kenya to be specific. And you know, with Kenya, we are known for the wildlife. So once you come to Kenya, ensure you have a game drive. And in addition to that, we have a strong nematology lab that's led by Professor Danny Coyne, who is under IITA, and also Dr. Solveig. So let me take you through my presentation. So I'll be presenting on a management practice for potato cyst nematodes, but we're focusing on resistant lines. We're calling them Shangi-Like lines, and I will explain why. So, and I'm presenting on behalf of Mellen NYABUTO, who didn't manage to come for the conference. Yes, and I'm Ivy NYAMBURA. So with potato is considered as the second most important crop in Kenya. And with that in mind, it's mostly farmed by small-scale holder farmers. And it's always cropped in year out, according to the rainy seasons that we have. And we normally have farmers, they don't do crop rotation. So that's kind of a problem for the potato industry. And over the years, we've seen that the yields are actually declining. And for some of the factors that farmers put into consideration while choosing different potato varieties is for short dormancy and potatoes which can actually cook fast and actually have a good taste. So with this, we have now the Shangi. Shangi is the most loved potato because it has zero dormancy. It cooks very quickly. It has a pleasant taste. And as you can see, when you compare it to different varieties that have been registered in Kenya, it's the most loved potato variety. So let me take you now to potato cyst nematode. So with potato cyst nematode, it was reported to be in Kenya in 2014. And we not only have it in Kenya, but we also have in other East African countries, that is in Uganda and Rwanda as well. So with us as nematologists, we wondered how could we actually control this menace. So in Kenya, we are having two species, that's the Globodora rostokiensis and pallida. And we can have the cyst, then in large quantities, we're having roughly 4 to 10 cysts per grams of soil. And actually, we're having them in 80% of the fields that we have in Kenya. So Kenya being now in the subtropical, we normally receive large volumes of rainfall. And normally farmers constantly farm potatoes. And the most loved potatoes I had mentioned earlier is Shangi. The most unfortunate part about Shangi is this, it's the most susceptible potato variety in Kenya. So that means that the potato cyst nematodes will actually increase in numbers while the farmers plant the Shangi. So we are having large volumes of PCN in different parts of Kenya. And we're not only having small cysts, but we're having giant cysts, which you can actually see with your eyes once you pull out your potato from the ground. And yeah, that's quite a big problem. So when PCN was reported in Kenya, coincidentally, the James Hutton Institute in the UK were working on different potato varieties that were actually resistant to PCN. However, in the UK market, these potatoes could actually, they were not marketable because they had a short dormancy. And you know, with the UK, once it reaches to winter, they don't plant anything. So they would have been written off. However, in Kenya, because now we always plant potatoes all through the year, the potatoes could actually be accepted in Kenya, and now we received the potatoes. So what we decided to do is we decided to take the different potato varieties, we're calling them lines, and then we decided to have them in different trials, and we tried to assess them for PCN resistance, if they could be accepted by consumers, and how they'll perform generally while they are affected by different diseases. So we managed to get roughly 8 lines. However, when we compared the yield for the different lines, as we compare them to Shangi, we noticed that the different varieties had higher yield compared to Shangi. And in addition to that, in fields where we have high PCN densities, we can actually see with Shangi, the PCN levels are increasing. But in other potato varieties, we actually see that the number of PCN levels are actually reducing. So with that in mind, a small recap of the different potato varieties, we can see that the PCN levels are being reduced, and then we are having higher yields, and then they have a short dormancy, and then they have a great taste and they cook quite fast. And from the pictures you're seeing, when you compare Shangi and the resistant varieties, definitely you'll go for the resistant varieties. It's actually big and nice. So we decided to have different farmers' field days where we could actually compare the different tastes. And we actually could see that, yes, farmers were accepting the potatoes, and we had them boiled, mashed, and in chips and in different locations. And the response and the feedback was the same, as you can see. When you compare that to Shangi, we had 6 out of the 8 varieties that farmers could actually like. And we have one variety called Manito, which is a registered resistant variety. However, it tastes horrible because I actually taste it. It's just not nice. So yes, the different varieties do work, and they're actually nice. So we decided to actually proceed on the 6 varieties. We did them in different locations to see how well they will perform. And we actually managed to only get 2 lines from the 6 varieties, which we could adopt them in the different geographical regions in our country, that's Kenya. And now with this year, we've proceeded on to the national performance trial so that we can see them registered, because yes, they're doing well and they're good. So we're hoping that we'll actually move this to other countries, these African countries that I'd mentioned, Uganda, Rwanda, and hopefully other countries as well. And in a nutshell, these new Shangi-like potatoes, because they're actually somehow similar to Shangi because of the taste and how they look like, and farmers love them, we're seeing that they are actually resistant to PCN. We are having higher yields, and they have a great taste, and farmers actually do like it. so. However, as I've mentioned earlier, PCN levels are actually so high in our country. And we are apart from the PCN, we are having blight, which is an issue. And we're trying to also assess how these different varieties will adopt in terms of the heat sensitivity, because we are living in the subtropics, we get to have varying temperatures compared to the UK, where these lines came from. And we are hoping that they will adopt properly in the different geographical regions. Yes, thank you so much.
So, good morning everyone. Firstly, I thank ICPP for giving me an opportunity to speak in front of you. So, myself, Dr. Chandra NAYAKA, I'm from University of Mysore, India. So, today I'm going to present on pearl millet. It is a future food for Asia and Africa. Its importance and its biotic contents and their management. So, why we consider as a millet as a future food? Because just I will give you overview because of this climate change and agriculture. It is mainly we have to acclimatize, depend on the changes in weather and other conditions like water availability and precipitation, etc. In India, it is contribute to agriculture to GDP is decreasing because of climate change, and yet large population are dependent on this world, this for livelihoods. Need to understand the impacts of increasing climatic risk and possible adaptation. So agriculture is a cause of climate change. It is one of the major reasons because because of agriculture. We also suffer from the consequences. The major adverse effects, impacts on climate change is increase in temperature, increase in rainfall pattern, decline in soil and water quality, shifting dynamics of insects, disease, soil flora and fauna, and intrusion of seawater on land, and biotic and abiotic stresses are arising. So we have to face a lot of challenges, mainly on genetic resource management, crop improvement for increased productivity, and genetic enhancement for high biomass, mitigating adverse effect of climate change, soil health, biotic stress management, abiotic stress management, and development of crop production technologies. So I'm actually focusing mainly on our Indian origin. So we have around 139.9 million hectares showing, out of which 57% of the total geographic area is rain-fed, and it provides 44% of total food grain production, and it is a livelihood to 50% of total workforce and 60% of cattle productions. So coming to millets, why millets? Because millets are weather smart, water smart, nutrient smart, and carbon smart, and also energy smart. And very important thing, if you eat millet, and we contribute very less price, and we are stay healthier and save earth. So compared to nutritive value with major crops like rice, actually it is 1.5 to 2 times more than rice, and fiber it is 40 to 50 times more than rice, and minerals it is 5 to 10 times, and iron is 10 to 20 times, and calcium 30 times more than rice. So it provides multiple security like food, fodder, health, nutrition, livelihood, ecological, whereas paddy and wheat only provide food security. So coming to water requirement, compared to any other crops, millet requires very less water when mainly compared to rice, wheat, and sugarcane and maize. So it is adaptive to climate change very easily. It is resistant to low rainfall and tolerate heat and low water requirement, and survive and flourish well under adverse climatic condition as well. So coming to global scenario, India is producing 80% of Asia's contribution on millet production and 20% of global production. the average yield is around 1229 kg per hectare. In India, it is 1239 kg per hectare. So coming to other continent like Asia is contributing around 162 lakh hectare, and followed mainly it is Africa. It is cultivated in 489 lakh hectare and production is around 423 lakh ton. And America, it is actually around 53 lakh hectare cultivation and production is 193 lakh ton. And Europe, it is cultivated in 8 lakh hectare and production is around 20 lakh per ton. So if compared to country-wise, India and Africa giving actually major contribution towards millet production. And the major millet we are actually mainly growing is pearl millet, jowar, and small millets. So in India, we are mainly depending on food in northern part of India is pearl millet. It is called commonly known as bajra, and followed by sorghum and small millets. So coming to pearl millet, why I am here is that we are mainly working on pearl millet. We have actually ICR center at University of Mysore, because pearl millet is the 5th most important cereal crop in the world and staple food for the poorest poor farmers, especially in India. We have actually place in northern part of India, we will get actually very less rainfall and there is no water, and actually the soil quality is very poor. It has the capacity to flourish under high temperature. The crop can grow up to 44 degrees centigrade. And it can grow in low rainfall and harsh soil, and low or no external input is required for cultivation of this crop. And pearl millet grain is highly nutritious. It is with 8 to 19% protein, low starch and high fiber, and higher micronutrients concentration than rice, wheat, maize and sorghum. So it is a C4 plant and is more stress tolerant anatomy, and the cell wall structure is made up of a tissue which are tougher for the pathogen to penetrate. So I will give you some example, like in wheat, there are nearly more than 200 diseases are reported. In rice, more than 350 diseases. In maize, more than 70 diseases, and sorghum, more than 65 diseases are reported. In comparison, this crop, pearl millet, is relatively less susceptible to pests and diseases. So about 50 diseases are reported on pearl millet. Only 5 diseases are major for economic importance in India and Africa. So the major disease what people are facing is downy mildew, which is caused by pathogen Sclerospora graminicola. It is a womycetes biotrophic fungi. It is a very serious disease in almost all major pearl millet growing regions. It is a seed-borne, airborne, and as well as soil-borne. And it can reduce the grain up to 75%. So from past 1 decade, actually in India and Africa, the millet farmer facing a problem with the blast disease. It is the #1 disease at present. It is mainly caused by Magnoportha griseae, and it is capable to reduce the yield up to 30 to 35%. So one more disease actually we are facing from past 1 decade is rust, which is caused by Puccinia substriata. It is the capacity to reduce the digestible dry matter of the crop. This disease actually mainly coming in later stage of the crop. So hence actually we are not observing grain yield disturb, but actually in fodder crop it is mainly reducing the yield up to 51%. So one more disease we are in major disease in pearl millet is ergot. It is caused by Claviceps fusiformis. It is produced one toxin, it is called ergotoxin, which is harmful to animals and humans. This pathogen can cause yield loss up to 70%. And one more major disease is smut disease. It is caused by Mycesiomyces penicillariae. In recent years, the disease has become more important in northern part of India and Africa, and it can capable to reduce the disease up to reduce the yield up to 30%. So recently in 2023, we have reported one more disease in India. It is caused by Bipolaris spicifera, and it is published in Plant Disease recently. And in 2022, we have observed and reported one disease. It's caused by Panthea stewarti, bacterial blight. It is also published in Plant Disease in 2022. So what we are practicing managing disease is that cultural method we are following, timely removal of weed and other inoculum sources can reduce the ergot and downy mildew, rust and blast disease, and deep summer plowing and destruction of crop residues and crop rotation is also going to reduce the disease incidence. And maintaining optimum plant spacing and regulation the amount of nitrogen fertilizers also reduces the disease like blast and downy mildew. So the main important thing is that managing diseases is host plant resistance in millets. It provides the most economic and environment friendly method for managing millet diseases. For food farmers, it is the only viable practice as they hardly use any other method of disease control in India. Recently released cultivars in India actually showing actually multiple disease resistant against downy mildew as well as blast. And in collaboration with ICRISAT in India, we have developed HHB67 improved recently in just 1 year back, which is showing actually multiple disease resistance against downy mildew as well as blast disease. So we are mainly using actually metalaxyl for management of downy mildew, and carbendazim and tricyclozole for blast disease, and bavistine for ergot disease, and mancozeb for rust disease. Apart from that, we are always looking for environmental friendly biologicals like Trichoderma argenum, Pseudomonas fluorescens, Bacillus subtilis, and we are used in different concentration as a seed treatment. So what we are doing at Mysore, actually we are planning to, we are actually developed a marker for disease resistance, which is highly sensitive, reliable laboratory screening techniques have been developed for hypersensitive reaction, histological, biochemical and molecular markers. So we are maintaining 8 pathotypes which are actually causing disease in different part of India. And we are used actually every year actually we are collecting the ooze spores and we are coating the seeds in vitro condition. We will evaluate the impact of the disease incidence of these 8 pathotypes. And we reported that the pathotype which we are maintaining in Mysore is highly virulence one. And we check the ooze spore production of these pathotypes every year. And also we will check the virulence index of these pathotypes by using different pulp millet cultivars. And also recently in collaboration with one of the institute in pulp millet, we have developed an artificial intelligence technology for monitoring disease incidence in field condition, and they have integrated with our genome sequence with their formula, and we found that 99% accuracy for identification of the disease in the field condition. So we have sequenced actually first time biotrophic homicides pathogen in any crop by using actually Illumina as well as PacBio. And this is the first report genome sequencing of Sclerospora graminicola. The overall genome coverage is 40x, and we have found the genome size is around 300 MB. And we have 65,000 genes that were predicted, and total of 38,120 genes were annotated. Also, we have sequenced the Magnoportha gricia of isolate, which is the highly virulent one, and we found that the genome size is 48 MB, and 3,150 genes were annotated, and the genes involved in pathogenicity, virulence, and effectors were annotated using pathogen-host interaction database. The genome is compared with rice Magnoportha gricia as well as wheat Magnoportha gricia, and it is different from rice and wheat blast pathogen. So we have developed actually a hypersensitive technique to screen the cultivars in vitro condition. We checked with resistant, moderately resistant and susceptible lines along with other hosts like sorghum and French bean. We found that the other host actually giving 100% HR reaction and followed by highly resistant one, and the susceptible one is giving actually less hypersensitive reaction. We have developed actually by using host seedlings, coleoptile and roots, and histological markers like lignin, callose deposition in the host cell after challenge inoculation with the pathogen, and we standardize the technique. And also we have developed a biochemical markers like for PR proteins as well as HRGPs, and we found that actually the marker we can screen we use this marker for screening resistant and susceptible lines. And we have isolated and cloned RGS, and that clone actually we have used for sequencing. From that sequence, we have developed a scar marker, and this scar marker can be used for screening in laboratory condition for resistant and susceptible seedlings. So apart from that, we are mainly working on disease management strategies. We are till now we are using actually metalaxyl as a seed treatment, and metalaxyl is actually a carcinogenic one. It is considered as a E group category in carcinogenic compound, but we have no alternative for management of downy mildew disease. So in India, especially in Asia and Africa, we are still actually looking for alternative for metalaxyl. So for that, we have checked with new generation fungicide like Flint, Amistar, and Strobilurine's compound in comparison with metalaxyl, and it as a seed treatment, and it is found promising controlling downy mildew disease. But metalaxyl proved actually it is a more effective than these fungicides, Initium, flipicolloid, paramycin, etc. So we are isolated more than 200 promising bioagents from natural condition. We are going and collecting the soil sample, and we are isolating the biological agents, and we have checked against the downy mildew disease, and we found that these biological agents are controlling downy mildew effectively. But if you compare with the metalaxyl, metalaxyl proving actually better than these bioagents. And apart from that, actually we have used more than 300 biotic and abiotic inducers as a seed priming agent, and we found that it is effective in seed downy mildew disease management. And we are using IDM module involving, and recently we are isolated spike associated microbiome, and we sequence that pathogens. So we are using silicon dioxide as a seed dressing agent, and we checked against whether these silicon nanoparticles can induce the systemic resistance in pearl millet against the downy mildew and the blast. And we demonstrate the nature of disease resistance by using QRT-PCR and biochemical assays. Like that, we are using different bioagents. We have purified actually lipopolysaccharide from Pseudomonas, and we checked against the management of downy mildew disease. And take-home message is pearl millet is adapted to severe drought, poor soil fertility, and high temperature condition, and it can be grow in without input or just actually 300 mm rainfall is more than enough for to cultivate of this crop. And the major income source for the 90 million poor people of the India and Africa. It is a most cost-effective strategy for effectively manage the disease can be through host plant resistance. And disease-resistant pearl millet breeding in India is relatively early, and field and greenhouse screening procedures seem promising. Work is currently being done identify eco-smart biologicals for pearl millet disease management that are less expensive, non-toxic, and can control biotic stresses and alter plant defense mechanism. Thank you for everyone. I'm sorry.
Thank you very much. We've had two presentations at the end, both very good. I want to say to our previous presenter also really lovely, particularly for a stand-in one. We have to go over to our last presentation. Would anyone in the audience like to ask one question for either any of the two last speakers? Okay, there's one. Then if we can get the microphone.
What kind of chemicals you use, and at what growth stage you apply the fungicides?
Sir, actually, mainly we are using metalaxyl because in pearl millet, none of the farmers are actually using for spray. So actually in India, whatever the chemical we recommend, actually we are using as a seed treatment. Because we have some part of India like Rajasthan and UP and Maharashtra, there actually farmers cannot afford any chemical fungicides. So if the company provides with the seed treatment, so majority of the companies will use apron. So for specific fungicide for any spraying and other things, they will not use, sir, in ergot. Mainly we are using metalaxyl as a seed treatment in millets.
I have a very quick question for the previous speaker. She mentioned some nice traits that the potato variety had, and one of them was climate smart. So I was just curious, how did you define how it was climate smart?
I'll say because the potato varieties are actually reducing the number of PCN levels in the field. So that means that we'll have less chemicals being used in the field, and that results to less chemicals being produced by industries. So yes, that's how I'll put it.
Thank you very much. And we're going to close the session. Thank you to both speakers, the last two speakers. And with that, our last session, we have a change in the program. So our last speaker couldn't get a visa in time. And we thought it's opportune for Serge to come and give us a little bit of an overview in terms of the state of food security. So over to you, Serge.
Yeah. And I was able to manage to get a visa to come here. So this talk is not in the program. And this talk is about your journal. You may not know this, but this journal, Food Security, is your journal. It belongs to you. It belongs to the society. That's the journal of the ISPP for the time being. There might be another in the future. But as of present, this is the journal that is published by the ISPP. And so it's important. I'm talking to you as colleagues. I'm talking to you as owners of the journal, because the ISPP is a collective, and the journal is managed as a collective. I'm working with 8 senior editors. They belong to very different disciplinary areas. There are 2 economists, there is a nutritionist, there is 1 sociologist, there are 2 agronomists, and I'm forgetting somebody. Anyway, it's a multidisciplinary team. We work as a team. Each team leader, each lead scientist, each senior editor is in charge of a publication line, on a publication strategy line, and we work together so that these strategies combine together and try to achieve that common goal of having a journal which is interdisciplinarily addressing food security. And so, for us plant pathologists, what is important for us to remember is that plant diseases affect all the components of food security. We hear a lot of the effect of plant diseases on production, which is okay. That is not incorrect. That is true. But there are aspects in terms of availability and stockpiles in the countries. There are aspects related to physical and supply chain. There are aspects of economical access to food. There are aspects of stability of food availability. Epidemics cause instability in food production. And there are aspects of nutrition, of course, which are critically important in some disease aspects. So all the components of food security are affected by any disease. On the top of the slide, you can see examples: potato late blight in the 19th century in Ireland, the wheat rusts today, the fusarium head blight in the Midwest today, the false smut of rice in Asia today, which is a major threat, the brown spot of rice, which is a chronic disease in South Asia, but also coffee rust in Central America today. All these things and many others, and most of the plant diseases we are working with are threats to food security. And so we have this kind of numbers, this kind of images where we can look at major crops in the world that are contributing to food security: wheat, rice, maize, potato, soybean, and we can classify them as chronic global, chronic regional, or emerging diseases. Lots of the literature and it's expectable, lots of the literature focuses on the emerging diseases, which is to be expected. The truth is that the vast majority, the largest fraction of yield losses caused by plant pathogen caused by plant pathogens worldwide is caused by chronic diseases, either global or local. And so this is our journal. I'm talking about something that you own. There are many journals dealing with food security. We're not the only ones. But food security from the ICPP was one of the first of the journals published on the topic. It was created 15 years ago. The theme of food security, despite the decreasing number of persons in the room, it's a fashionable topic. It's fashionable. People like to write food security in their manuscript, somehow. But it's often a pretext, let's face it. Aside from that, it is a very extremely active field. As I said, with sociologists, economists, agronomists, but not so much among the plant pathologists in reality. And that's because it's involving science, but also policies and politics. It's a very volatile subject. One of the most frequent keyword, and I'll be back to this in a minute, in the last few years was COVID. COVID caused a tremendous level of volatility in the food security and the food insecurity across the world. It's an emotional subject as well. And so food security is a science journal. We deal with science. We do not deal with emotions. It's difficult sometimes. And so we are looking at truly interdisciplinary fields of research. We're looking at biology at all scales, from the molecular, and this is important, from the molecular to the field and the global scale. And so when a plant pathologist works at the molecular or at the gene scale, or whether that plant pathologist works at the field scale or at the global scale, and there are some, all these scales are relevant to food security because they ultimately will contribute to food security. Otherwise, plant pathologists in many cases would not be doing that. So there is one thing one message which is very important, which I would like to convey to you, especially to the young people, which is that what plant pathologists, what we do, what you do, is important. It is important for plant pathology, yes, but it's important because it has an ultimate purpose, which is food security, in very many ways. And that should carry you. One should be proud to do this. It's a matter of pride for oneself. It's a matter of being more happy of doing what one does. Oh, it goes with responsibility, of course, and it goes with ethics issues. But it's important to know that. We don't do this for no reason, and we don't do this for money. If we wanted to earn more money, we wouldn't be scientists. We do this because we think it's important, and it's important in many cases because it's about food security. And there are other things. It involves economists and sociologists and natural resources and environmental physics at all scales of geography. Oh, there is a senior editor for geography, economic geography, and their applications, nutrition, agronomy, and etc. So what I've been doing in the past few months, checking my watch, is that I've been asking myself a few questions, and questions that databases from the web or from the publisher cannot provide me, even though as editor-in-chief I have access to some statistics and thingies that the publisher provides, but that's not good enough to me. My question starts like this: what are the broad themes that are addressed in submissions to food security? Not what is published, what is submitted. Where do the submissions come from? No, not which lab or which country. Which terrain are they coming from? What do they look at? Which part of the world are they looking at? Okay, if they are looking at the world globally, then I will come back to the country where it comes from. Can we identify a classification of submission themes? What is submitted, not what's published by the journal. And if there are dominant themes, why is it so? Why is it so? You all know this. We all know this. There are fashions in science. me, I am very worried. I'm very afraid of fashion in science. And so I want to know why these things are dominant. Is that justified by field truth, in other words? Is that justified by the reality? And ultimately, what is it that we are overlooking all the time? And this is why I would love to have feedbacks from you. What is it we overlook? Is it your journal? So my hypothesis were that there are some obstacles that to food security and and obstacles to remove food insecurity, which are stronger than others. I'm assuming that some obstacles are temporary, whereas others are long-lasting. Some obstacles may be looking like minor, not so very important, and persistent, but persistent. Whereas other may be big, but not so persistent. And so we, with my colleagues, with the senior editors, we have to navigate an editorial board, an editorial policy of the journal across all these elements. So what I've done is to look at the shape of submissions, and to do this, I use approximately 2,000 submissions to the journal between 2020 and 2022. We receive approximately 800 submissions per year. And we publish approximately 15 by 6, so nearly 100 papers a year, 100 articles a year. And so each of these items, each of these 2,000 submissions are represented by a submission code, the type of submission, the title, the author, that's confidential, the country, the world region, where the research is conducted, where it is focused, and the outcome, whether it was declined or published. So I used word analytics thingies. I used different types of statistical methods. All of this is about words, and so the metric used is a chi-square distance. So just to give you, because that's your job, you need to know this. On the left-hand side, on A, are the countries where the submissions are coming from. And, well, no surprise, India and China are big suppliers of submissions. But as you can see, Ethiopia is a very big supplier of submissions as well. So I see Ethiopia as a powerhouse in research on food security today. And what I see in sub-Saharan Africa is that there are not so many other countries. There is no so much. There are other few lights in sub-Saharan Africa, but most of sub-Saharan Africa is not very active, and that's a concern. Why? Because even though sub-Saharan Africa is less than 1 billion people today, its population growth is the fastest in the world. And the other part of the world, which is a very major source of concern, is Southern Asia. Southern Asia, that is to say Pakistan, India, Sri Lanka, Bangladesh, Nepal. These countries represent one of the most populated part of the world. This is still the largest number of people that are undernourished, malnutrition, and hidden malnutrition. And it's the largest population of people who suffer from hunger, and climate change is hitting South Asia far more than any part of the world today, and it will in the future. And then on the right-hand side, you have what's published. So the distribution, the densities you see are resemble in part to the figure in A, but there is a change in order because some papers are more easily accepted in some parts of the world than others. So these are the fractions, accepted, decline. It varies across regions in the world. It's not, okay, there are variations. Sub-Saharan Africa is the largest contributor of submissions. It's a log scale on the left-hand side. So it means that SSA is very, very big, much bigger than the others. South Asia comes next, etc., etc. There are differences. You can elaborate on this. I wanted to think about the scales, the rural, the urban. It's important to know this. More than half the world population became urban in 1996. We all speak of the farmers, Yeah, but the largest population of the world lives in cities today, by far. And the largest proportion of poor people live in cities today, by far. So we keep on focusing on farmers, that's fine. But food insecurity, that concerns urbanites as well. And this is important for us to remember. Food has to be accessible to the poor people living in cities, because that's the majority, after all. And so I looked at those things, and then who is looking at what, producers or consumers? So that's back to what I was saying. Some countries, some parts of the world are focusing more on the consumers, such as East Asia or MENA, Middle East and North Africa, another area of concern. Booming population, tragic climate change effects. And Sub-Saharan Africa, as expectable, is focusing much, much more on producers than on consumers. Why? Because that's where the largest fraction of people working in rural areas is living. South Asia resembles that a little, despite its megacities. And then I was very much focusing on the nutritional aspects as well, and I wanted to, and that is connected to the topic of, in terms of actors, children, women. More than 60% of the agricultural production in the world is a fact of women, not men. 60%. And they do all the rest. And so, well, you can see that children are quite often addressed in Sub-Saharan Africa or North America, where food insecurity it does exist, and food security is an international journal. It's addressing North America, Canada, the USA, Western Europe. It's looking at all the countries in the world in the same way, different problems. So what we see is that in countries from Latin America, we don't get publications, submissions dealing with children, which is interesting. Most of them are dealing with women. And then let's not enter into this. This is the cooking. This is the kitchen. These are the groups of words, the word analytics thingies, which I did. And I came up with a number of, maybe, let's say, 5 key groups of, maybe you can see the pointer like this, if I do that, can you see something? So food production is a major theme. Change, scientific or technological. The economy, with all the elements of economy, many scales of economy. Economy of the household in particular is very important, but macroeconomy as well. Crisis. I removed COVID from the database. I removed COVID because I knew that we would crash in everything else. So let's get rid of COVID for a while. And let's look at the other things. So otherwise, there's nothing to be seen. I could show that. But so crises are very important. And nutrition. Nutrition is a mounting subject, is critically important in the world community. People at long last realize that, yeah, you just don't need calories to be, okay, you need calories to stay alive, but you need something else in your diet in order to have a healthy life. All right. But there are people that still don't have the calories, though. There are many of them. So these are the correlation. So previous slide was, where are those different things addressed more than okay, let's pass on this. This is whether there are associations between the things. So nutrition is very much associated with food production and not so much with the rest. Crisis is associated with food production and economy. Economy is not associated with those two, and economy is dissociated with nutrition. So there is a clear disconnection between economy and nutrition. Interesting this, funny. But economy is very strongly associated with crisis. Of course, economists, they love crisis because that makes papers. But no, I have nothing against economists. And they are good friends. And food production is associated with change, technological change, socio-economical change, things like that. And so this is a map. Please look only at this one. This is a map. This is a geographic map, but it's not in meters, it's not in kilometers, so it's not in Euclidean distance. The map is on a chi-square distance. So on this chi-square distance, we see that here are our 5 clusters of the 5 theme clusters. The crisis up there, the economy here. Very far away from crisis, we have technological and social, socioeconomic change, and next to it we have food production, sort of. And then in another part of the galaxy, you have nutrition. So look at those things as galaxies in a global planetary system. These galaxies contain a number of worlds, and those number of worlds are cited a number of times, and therefore their inertia is different because their weight is different, but they also are more or less far away from this center of this galaxy system. And so crises have a very, very strong inertia. It's proportional to the weight of this thing, of the mass of this thing, and the square distance to the origin. So there's the economy, nutrition is also very, so these things are having more or less similar inertia. When you rotate them around this center of axis, they have, and so if we look at the other themes, the blue thing is here, no need to look too much at them because they are close to the center, meaning to say they do not bring much more information. Their inertia is less. In other words, they are pretty well described by the other 5 black dots. If we look at, okay, that is critical, are we biased? So despite the way I behave and despite the words I use about economy, you can see that we do not have bias at food security. We don't. We have acceptance and decline approximately at the center of this galactical system. And that is, of course, because we have outstanding senior editors in economy, together with associate editors. A very large fraction of the submissions are coming from the economists. And we have no bias either for any region in the world. By the way, look at what regions of the world are indicated here. There are many, but some of them are North America and Australia. We take them all. We take everything. Everything is good. Nothing is out of scope when it comes to food security. And so this is probably maybe my last slide. I know it says 19 out of 20. I don't know what is the last one. Whatever. Anyway, same map. It's the same map. So we have still the crisis thingy here, the nutrition galaxy there, the change galaxy here, the what was it? Food production galaxy there, and then the economy. One interesting feature of this map is that there is another galaxy hidden behind, which is the technological background, the infrastructure of food security. And here we have supply chain, which is one type of infrastructure. And here somewhere here, you have farm, which is another type of infrastructure for food security. So this galaxy is cutting across everything. Is hidden behind it. So we have keywords such as resilience. Resilience is somewhere between the center of this galactic system and crisis. Makes sense. And we have sustainability, which is on the axis of technological change and food production. So well, okay, it makes sense all this. We have the key actors. We have consumers and the public sector, consumers and I don't know where the producers are. I forgot to place them. Anyway, consumers are here. The producers are necessarily somewhere there, balanced in between the origin of axis. And we have the two others. We have children, the child here, very close to nutrition. By the way, nutrition is about words such as dietary, health, nutritional, iron, which is a major issue. More than half the women in India are anemic. Anemia, when you're pregnant, is a major cause for mortality at birth. It's a major issue, this. And so the previous speaker was right to mention that millets are a source of iron, much better than other cereals. The problem of millet, though, is that millet doesn't produce as much as rice does. Anyway, so this nutrition thing is there, and it's close to chocolate. But why on earth is women here? Well, women is here because it's close to production, of course. Women are not concerned in the articles by just only nutrition. They are concerned also, and luckily enough, by production, and that's fair. So that's the shape of the journal. So this is the landscape. So what I presented to you is the landscape of mindsets of the authors who are interested in food security. It's not what we do. This is what we get. You got what we produce from these 2 points: reject or declined and published at the center. So we're approximately at the center of this landscape, which is fine. So no bias. But there are concerns. Me, I have concerns. My one concern, first concern, is sociology. Sociology in food security related themes seems weak. And in plant pathology, sociology is extraordinarily important. Sociology is related to adoption of technology. And so sociology is associated with the adoption of breeding products or new techniques or whatever. Sociology is extraordinarily important. We are not good there. We don't get sufficient submissions. Biodiversity. Food security is at the center of so many threats. So one of them is the collapse of the global biodiversity. Another one is the climate change crisis that we are starting to face. Another is related to the economic fragility of the financial system globally. And we've seen that happening time and again, and we'll see that happening again in the future. It's unavoidable. And then there are accidents which are likely to happen again, such as COVID and things like that. So me, I don't see, maybe you think different, but me, I don't see as food security as a theme in its own right. As a doctor, I would see it as a symptom of many things that are going wrong at the same time. And this is interesting for us plant pathologists, because symptoms, that's our stuff. Well, together with doctors and veterinary vets. And so I'm concerned to not see biodiversity. We see only 60 manuscripts, which is only 24% of the number of COVID submissions. And what I do think, and this will be followed up by the session on global plant health assessment. The concern I have is that we still see the dilemma between food security and a surviving biosphere. There still seems to be a dilemma between those two objectives, and it should not be so. It cannot be so. We must, as scientists, I think, find a way to address the two at the same time, because otherwise there's no future. And I am very worried also to see so little manuscripts submitted on that deal with climate change, but that's expectable. That's reasonable. You can reason this that way. There are many, very many journals, and excellent journals, that are dealing specifically with climate change. The thing is, though, that climate change specialists, climate change scientists are not so very interested in food security. So I would like to attract them more. We would like to attract them more. And with this, I'll be glad to answer any question you will have. And
thank you very much, Serge. That was thought-provoking. I know we're at the end of our session, but I'm sure there might be a question or two. Anyone? Would anyone like to ask Serge? Yes, there we have one hand. Can we get the microphone?
Thank you. That was very interesting to listen to and learn from. I'm just wondering about the Francophone part of West Africa. Has a lot of Francophone speakers, and I think a lot of them are not able to get into journals like this because of that language barrier that they have. Is there any thoughts on addressing that?
Well, me, I'm Francophone too. I've worked in Africa 10 years. And I know Africans. I know that people in sub-Saharan Africa are extraordinary skills in languages. Anybody in Africa speaks at least 3 or 4 different languages. I don't see submissions that are coming from sub-Saharan Africa, especially from West Africa. These submissions are good quality in terms of English. No. There is no. I don't think that's an issue. I don't think that's an issue. I think that there is no barrier. Besides, we don't care. If the English is not so nice, it's not ground for rejection in any way at all. What's important is the value of the submission, the intrinsic value, whether it's in the scope or not, whether the stats are okay or not, whether it's reasonable or not, I mean, whether that makes sense or not, whether that falls in one of the publication strategy lines of the journal or not, but not the shape, the style of the submission. Yeah, well, that's why we are, no, let me rephrase this. We are editors, so we receive manuscripts. Our goal is not to read, our goal is to publish stuff. And so and this is why we are slow. Food security is a slow journal. And I get people, the publisher is cross at me all the time. We are slow because we want to. We are slow because we review stuff. Good reviews take time. We do it. And reviews are looking at the language also to some extent. And then when a submission is past the process of review and things are okay, then it will be cross-reviewed by English editors. So no.
Well, if this is one more final question, yes, I think I saw that hand.
No, we can stay
Lise.
longer. It's food security. So I'm not hungry, so I don't know you. Me, I'm happy to talk. Are you hungry? You hungry?
No, never.
Oh,
No.
okay.
Always thirsty.
Yeah, have some.
But I am food secure.
So my question is around sociology. What would be your opinion on what elements of sociology you would like to see as the editor?
I'm not sure to understand your question. Right.
So one of the key concerns was sociology,
Yeah.
the human component,
Yes.
and the the ecology,
Yeah.
biodiversity.
Yeah.
What elements do you think are going to, what trends, I don't want to use the word trends, I don't mean it in that fashion sense. What do you think is critical from a sociological point of view for food security?
To me, well, I'm not a sociologist, see?
But as a plant pathologist, we have an element of sociology in our
I'm
discipline.
very glad to hear you say that. But still, I'm not a sociologist. And so the key word I would use is human capital. Is human capital and so that's human capital at different locations in different scales. Human capital at the household scale first, and then human capital at the field scale, and human capital at the community scale. These are the things that to me are then human capital in the urban settings. I'm very insecure there. I don't either. It must exist. It must be very important, but I don't know the subject.
Okay, I think it's a good time to end. Thank you very much for that input, Serge.
Thank you very much for coming.
Yeah.
Thank you.
And just 2 more things before everyone jump up, run out. I would like to ask the speakers just to stay behind for a quick photograph. I also want to invite you, if you're interested in becoming part of the food security future landscape, to please just stay behind for a minute. We just want to get your details. And we're particularly encouraging the young next generation to join us. There's a few platforms, but you're most welcome to contact me anytime or Serge.
Yes, you'll be kidnapped.
And we'll hijack your mind, body and soul. And then, so please interact with us as much as you can, join the commission, support our journal. And then I'm going to end the session just in memory of Dr. Richard Strange. And for those of you who had the privilege to know him, he was a man, a wonderful, inspiring man. He worked in the old Rhodesia, where he did his PhDs many years, but he was a true African and a person working so hard in so many countries all over the world. And he was also instrumental in starting our journal and also the task force on food security, which was taking up Norman Borlaug's challenge, not only to discuss food security, but to also do that. And this today, for me, was a session about doing food security. Yesterday, we heard about the theory and the big dreams around food security. Today was about doing food security. So thank you very much for your time, for coming and supporting us. And please, tomorrow, Thursday, there's one more food security session. There's a post-harvest waste and losses one, and do support us. Thank you very much for your time.